So, yeah, I think the reason most of you are joining up is either you've had bad experiences with mites and diseases, or you have no experience with mites and diseases because you're a new beekeeper. So what we'll do is we'll go through the ABCs of basic pest and disease management. We're going to talk about bee stressors, because usually when bee stressors start happening is when these diseases start popping out. So best defense against pests and diseases is good nutrition. So adequate nectar sources or sugar syrups if you have to, and then pollens or pollen substitutes. So a lot of like EFB and the PMS, especially if there's no mites, usually some sugar syrup and some pollen patties will help make it go away. You take away the stressors, and a lot of times the diseases go away. Another key one is healthy, productive queens. A lot of times to flush diseases out of a colony, you need a queen that's really producing well, a lot of young, fresh, healthy bees, and that usually comes from the queen. So a queen is really important. So I find that year 2 to 3 is where my queens start falling behind, and where I may start seeing some queen issues. So now my goal is to requeen every year or every 2 years. Another big one is beekeeper education. So this is the key one actually, learning to recognize potential issues, so before they happen. So being proactive, but also learning to recognize actual issues. And that's where you react and you do something to try to fix it. Every time you go in your colony, you need to start getting a feel of the sounds of that colony, the hum, the activity, the smells, the sounds, because it tells you a lot about what's going on in the colony. So for example, a couple of days ago, so 4 or 5 days ago, I went into a colony and the bees were lethargic, weren't really moving around at all. And I knew there was something fishy with that colony. So I did find a supersedure cell, hard linear brood, some drone brood. So basically it was just the queen. I was having queen issues. So luckily somebody was bringing in queens and I was able to get a queen, and I went through an introduction, and today I released the queen into the colony, and in 5 days I'll check again to see how she's doing. But usually you can cue in on a lot of colony issues. You might not know exactly what's wrong, but you'll know something is wrong. And then the other key one is I try to get people to share their findings or to ask for help, because we've all gone through it. Like I've dropped a frame full of bees by turning, and then I threw it like 6 feet, 8 feet away, and all the bees went flying everywhere. We do things. I've had bees, I've had mite bombs, I've had colony collapse where the whole colony collapses because of severe mites. So it does happen, and the key is to learn from it and to share and to try to grow from it. Okay. So the key to knowing about diseases is to know a bit about anatomy, a bit about biology. So because we're going to focus on some brood diseases, but mostly Varroa, the key is brood. Okay, so for Varroa to multiply, they need brood, they need eggs, and then they need a capped sail for the mother mite to lay her eggs. And without that, basically she's just hanging on to bees, and then they're at risk there. So hard to see, but this chart on top here is the bee population, the red line, and then the yellow line is your brood. So hence the reason why we're lucky up here is we do get a fairly decent brood break, where basically our queens aren't really producing brood. And then during the winter, as our bees die out, just from normal winter mortality, Mites are also dying, but if they do survive, so they're not multiplying, and that's the key. Is over winter the mite population isn't multiplying; it's just at a standstill. Unfortunately, we can't really treat like they do down south. A lot of places down south, they'll do a oxalic acid vapor treatment in January or December. But here it's too damn cold, and you disturb the cluster, and we can't really do that. So it's just something that we can't do. In spring, so what I'll do is I'll just go through the basic bee cycle, and you'll see that, for example, spring is the best time to do a first treatment because the mite population is at its lowest. So for those bringing in packages, it's a beautiful opportunity to get rid of most of your mites right as you start, okay? Because there's no brood, and basically there's no brood. And because there's no brood, all the mites are exposed. So a typical package, I think it's a 3 to 5 pound package, will have about anywhere from say 50 to 400 mites. Okay, so we're all used to the COVID world and exponential growth. So for every mother, she'll lay 2 eggs and she'll raise 2 daughters. And then those 2 other daughters will raise 2 other daughters, like mites. And eventually that population goes exponential, and then it tends to peak in fall, pretty much when our brood rearing stops. So all the mites get exposed, but all our winter bees, basically late summer, fall is when you've got the most mites in your population. So it's affecting and it's stressing out your winter bees. Okay, so we got spring, raise new brood. Early summer, the colony is growing, so they're collecting pollen and nectars. So we get into a bit of swarming. Midsummer, pretty much it's about maintaining colony. Again, they're collecting pollen and nectar. So, and they're starting to ripen and packing away some honey. The key with early summer, so the thing about swarms is you could do splits. So we're going to talk about brood breaks. So making a split is another natural way of controlling your mite levels, especially for a year or 2 colony. So late summer so is where we start producing winter bees. So again, you'll see it's pretty common, collect pollen and nectar, and they're starting to prepare the nest for winter. So as this is where the mite levels are starting to go to their extremes and they're peaking. So, and it coincides with the production of winter bees, which puts them at risk and stresses them out. And then in fall, the bee population, so the summer bees start dying out, and the mites have a knack of not dying with those bees, so they just jump onto the winter bees. So now your mites per bee is increasing. Okay, so, and mites can feed on bees, so they're not sucking the blood, they're actually feeding on the fat body, which is like the liver of the bee. So they're actually feeding on our winter bees and stressing them out. And I'll go through some of the, I guess, the functions of the fat body and how important that is. And then we start over, and then early spring, the cycle starts over. Okay, so typically I have the lowest levels, which is normal, in early spring. As you get better at overwintering, and you overwinter, say, a colony with like a double with 12, 14 frames of bees, you'll notice that your mite issue will actually increase. Because you have more bees surviving, it also means more mites are surviving the winter, and chances are your colonies are brood rearing earlier because there's a big healthy colony there to keep the nest warm. So I find big colonies start rearing brood in late February, sometime in March, even without pollen patties. But a weaker colony will have to wait till mid-April to May before they really start brood rearing. So This here just shows the cycle. So this red line is the brood rearing cycle. So it's the temperature in the colony. This brown line is the temperature outside. You can see this is May, and it's a full cycle. So May to pretty much August, so 2 seasons. So this is the period that, in this period here is where the mites are really, so it again, growing exponentially. The queen stops laying, they're dormant for the winter, meaning they're just clustered, not producing bees. So the reason I know they're not producing bees is I've monitored the hives, and I know that typically they won't start rearing till sometime in late January if this was a really big colony. So there's a bit of brood rearing going on, but it's minor till March. And you can see first natural pollen is for us here in the Yukon, in my location, it's late April, early May. So wherever this line is flat, it's prime mite raising temperature. Okay, so you can see most of the summer mites are growing. So we all know, or hopefully we know, so mites are attracted to drones, probably through smell, but also because they also have the longest egg to emerging cycle. So a drone is 24 days, a worker is 21 days, and a queen is 16 days. So they get almost 2 rounds of egg laying in a drone versus 1 and a bit for a worker. So they will lay eggs in worker cells, but they seem to be attracted to the smells of the drones, and chances are it's got to do with the longer brood rearing cycle. So one of the techniques I'll talk about is actually putting a medium frame, because we've got these these green frames, these drone frames that people talk about. They're good, but up here again, our season's really short, and I guess the research coming out that because these mites carry viruses and then they transmit them to the larvae and the bees, we tend to freeze these frames and then just put them back in the colony for the bees to clean out. Because bees like to recycle protein, so a larvae that's dead, a lot of times if it's still viable, they'll eat it. They'll cannibalize the larvae if they're low on protein or if there's available protein, instead of discarding the larvae. So I guess it's looking like one of the virus transmission points is actually through some of these green drone frames. Okay, so but we'll talk about a medium frame, how you just put it in a deep, and they'll draw out some comb in the bottom of these medium frames. And then you just scrape it off once the drone comb is capped and there's a bunch of a drone in there, and you can open up the cells and check for mites. Okay, so next one, bee genetics. There's a lot of, I guess, when you are buying your queens or your packages, sometimes you get to choose the type of queen. A lot of times up here we pick carnies because of better winter survival, but there are also, I guess, I think in the US you guys have access to the Purdue, the ankle biters, and so Varroa sensitive Hygiene. So there's a few breeds that have been raised to basically be very hygienic. And this is also good for brood diseases. And I'll show you the next slide. But these are traits you look for. I look for docile bees. So if they're easy to inspect, you will inspect them. But if you have very pissy bees that are aggressive, chances are you might push off some inspections. Hence the reason why you may not control your diseases very well. So It's important that if you do have very defensive, so bees that attack you, it's probably time to change that queen. Swarming, like I won't go through all this because it's a whole different workshop, but for myself, swarming is good because it allows me to do splits. So by doing splits, I'm breaking the brood cycle, which helps me control my mites. So, and yeah, I'll skip over the rest. So over here on the right-hand side, this is an example of where you put a tube on your brood frame, and then you pour liquid nitrogen. And then over a 24-hour period, the bees would uncap and clean out these cells. And how quickly they do it, or how much they do it, indicates how hygienic these genetics are. So for example, this is from the Be Informed website. So I put the link here. So this is poor, this is good. So a lot of queen breeders, they test for this. If they don't do this, and they say they have Varroa sensitive hygiene or hygienic behavior, and they don't do this, then it's not true. Okay, so when you are buying your queens, please ask the breeder to make sure that if this is one of their sell points, that they actually breed from queens that they've tested. Okay, because and it's not 2-3 generations off. So this is a yearly test that they should be doing on all their breeder colonies to verify how hygienic those queens are. So you could ask them to say, hey, can you show me your stats? These are, again, the same traits. Like I talked about. So again, ask the breeders that claim Varroa sensitive hygiene, ask them for their test results. And then you'll see if they actually do it or not. So part of disease management is doing actual inspections. So if you're never in the colony, it's kind of hard to know if there's any problems in there. Again, you don't need to have big, really long inspections. For example, 5-10 minutes with experience, you can probably inspect half the frames and get a really good indication of the general health of that colony. And I'd say here I do it 10 to 14 days, but when you're first starting off, I'd say every 5 days is fine, 5 to 7 days, because you need to get really a good understanding of these colonies. And in the next slide, I'll show you a trick you could use so that you don't over manipulate some frames. So if you're going to do it every 5 days, if you always pull out the same frames, you might stress the colony out. So what I started doing on a few hives is I have a big Sharpie and I actually write what's a honey frame. So if you can see the HC, so an empty comb. So drawn comb, brood, 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 comb, comb, honey, empty. So I like Sharpies because probably after about a month, you won't be able to see it anymore. But what I do sometimes is I might put the date or a next on this frame, and I'll do my inspection in this direction, and I'll inspect 5 frames this way, week 1. And then the following week, I'll inspect this way. So if you just put a Sharpie mark here and have a code or something, the date that you inspected, then you know to start from this side. And that way, in a 10-day period, you'll have seen all 10 frames in that box. So keep a Sharpie and you can do whatever you want with it. There's no right or wrong. So here, for example, are things that sometimes I have a piece of duct tape on some of my colonies on the lids. And I put so this is a Yukon Queen from 2018. So I just put YT there. Most of my queens come from BC. So I just say BC 2000, or sorry, BC 20, Queen Right, seams of bees. So if I might have 7 as bee, so how many seams of bees? So for example, in this box here, I'd say this is a partial, but 1, 2, 3, 4, 5, 6, 7, 7.5 to 8 frames of bees. Okay, so frames of brood. So there's 3 frames, and frames of honey. So there's 3 frames of honey, and a date of inspection. So I would just use a piece of duct tape, and you write it on there with your Sharpie, and it's all good. And for example, I inspected this colony 10 days after this, and these 2 C's now were bees. So they had brood on them. So when I did my assessment, now this colony went from 3 frames of brood to 5 frames of brood. And so this colony is heading in the right direction. So I haven't really talked about diseases yet, but if you keep healthy colonies, diseases aren't an issue. Okay, so first sign of chalk brood, I use screen bottom boards with inserts. And I think I talked about this in that first talk, but these are chalk brood mummies. Okay, so if I see this on my screen bottom board, I know that I have chalk brood, and then I'll check the frames just to see how bad it is. If it's minor, I'll just throw some feed on, maybe a pollen patty, and I might put an extra box or change the boxes to fresh poly or a fresh wooden box, just to give them a new hive home. So, because sometimes humidity and cold influences them. Okay. Oh, there's somebody in the waiting room. I think they're good. So let's see. And the reason I really like my screen bottom boards is for mites. Okay, so natural drop, you can see if you have mites, and I'd say that's the extent of a screen bottom board. Is a natural drop will tell you I've got mites, yes or no. Okay, it won't tell you how many, because in the last 5 years of monitoring, I haven't seen a relationship between how many mites and what the natural drop is. And natural drop is a dead mite or a groom mite falling off a bee and then making its way down to the debris board below the screen bottom board. And technically you would clean the board off, insert your insert, and then come back in 3 days and count how many mites you find, and then divide it by 3. And that would give you a drop for 24 hours. Okay, so I do an OAV, and then I count the mite drop. And then I get a higher count, and it tells me more. So for example, this was after a oxalic acid treatment. I think I might have had 5 mites per 24 hours. Not even that, maybe 3 mites. And you can see I did a treatment, and in here, ballpark, there's a couple of 100 mites here. And year 1, probably not an issue. Okay, so the colony will probably make it through. But year 2 is where you start seeing issues with viruses and potentially them not overwintering. And sometimes it gets so bad that it starts affecting your brood. And that's where PMS comes in, so parasitic mite syndrome. So that's why if you get a new nook or a package, usually mites aren't really an issue until year 2. Okay, so we tend to hold off our treatment till the first fall or the following year. And then we wonder why we're having such trouble ramping that colony up. So my recommendations for most folks is treat when you get that package. So install them, give them a couple of days to get comfy, maybe a week, because they won't have time to really drought and cap their cells, especially if you're starting with like new equipment with no drawn comb. You've got a good week to 10 days before they start really capping anything, if even that. You've got more time than that. If you've got drawn comb, the queen might actually start laying almost right away. So you've got, I'd say 5-6 days before they start capping cells. So, or 8 days. So it's important that you don't wait too long. So other inspection things, again, somebody was asking me about where do I decide to go from a double or a single for wintering? So basically, if this is a lot of bees, if I get this times 2, it's a double. If I got this in one box, but the other box, it's not side to side bees like that, then I may do a single. But I want lots of bees. So and the other thing I start doing is taking out some of these side frames and using follow boards, putting styrofoam and wooden pieces of wood to reduce the volume. So I'm looking for density, so a lot of bees in that space. So if you've got too many for a single, so sometimes I'll start removing some frames and filling that with insulation just to condense that colony down. So again, chalk brood, this is dysentery, not necessarily nosema, and swarm is good, and seeing lots of pollen come in is good too. So we're going to start getting into diseases now, but again, critical, I talked about it last time, so pollen is critical. So you do want your population to grow, because this is where you'll lose your colony. Okay, so there's a lag on there, but our low, for example, for me, I'm in the low right now. Today was the first sort of spring day here where it was fairly warm, and the colonies were on crocuses, collecting crocus pollen and soap berries and nectar there, but it's been really cold and wet here, so they haven't really recovered. So I had to reduce 2 colonies down to 6 frame nooks, just because they weren't able, they're not able to grow. So I'm condensing the nest down to bare minimum in a nice insulated nook box to keep them nice and warm so they can grow the nest bigger. I'll get them really dense and then I'll put them in a single. So sometimes that's it's a way of kickstarting it. So the challenge with that is, this is also where this is your start number for your mites, whatever the number is here. So if it's 10 mites, I found, I'll get to that soon, but I found that if I do an OAV and I see 10 mites or more, I keep running the treatment every 5 to 7 days until my count is below 10. I've got a couple colonies right now up in the 70 range, and the other 6 are below 10. So 2 of them have 0, 3 of them have 0, and 2 have like 5 or 6. So it's not too bad. So I'd say I've got a quarter of my colonies that I have to do retreats on OAV. So I'll skip that one and I'll skip this one. But just so you know, these are tricks. This is that spring ramp up. I took it from Randy Oliver's site, and here's the source. And it's everything that grows your numbers and everything that reduces your numbers. Okay, so what you're trying to do in spring is you've got to give them everything they need to basically grow as a colony. And the less stress they have, the better. And again, this is just a trick I use to boost weaker hives. I've got a couple of boomer hives that have extra resources that I may steal some nurse bees from in a brood frame and transfer them into some of my weaker colonies. And that's called equalization. And this is the time to do it before we get into our first nectar flow. So again, we have really old bees at the moment. A lot of, I guess, if you're southern Alaska, it's not too bad along the coast. because you've probably got 2-3 weeks to a month on us weather-wise. So your colonies are about a month ahead of ours. But again, so around winter bees, and what kills them is Varroa stressors, nosema, some other diseases, cold stress, metabolic aging. So a lot of these other diseases, cold stress and metabolic aging, there's a direct relationship to the number of Varroa. So you can see that other than nosema, probably 6 of these factors are actually related to Varroa. So if you do nothing, then chances are your bees won't really do well. And there's more, you don't have to use chemicals. If you don't use chemicals, especially hard chemicals, so like miticides, you can use soft chemicals, certain essential oils, OAV, formic acid's a bit tougher on the bees, but they don't leave any residue. So there are ways of managing mites without putting chemicals at all, but it will require more of your time, and we'll cover that in a bit. So again, lots of bees is good. And then we're into diseases now. So I think, so yeah, the key is stress. Keep the stress under control. So it's better to prevent stress than to cause it. So again, poor nutrition. Early in the season, don't be shy about putting pollen supplements. I use substitutes, supplements here. Because most of our pollen patties have pollen in them, real pollen, 15 to 55%. And we do it until they stop taking it. So I added some yesterday because it was cold and wet for about a week. But hopefully the weather changes and the willows start producing again. So chances are I won't need pollen patties from now on. But it's there, and if they don't take it, they don't take it for $2. So I put a half pollen patty on the colony. So for $2, I think it's worth the investment. I'll do a talk specifically on wintering, but I guess for now we won't worry about that. Mite population. So again, the peak. So IPM. So what's that again? Integrated pest management. So we'll give you a couple of tricks relating to mites. So monitoring, so you need to know how many mites, if you have mites. And for myself, I just use OEV twice a year. And if I stick to my routine, it works. If I don't stick to my routine, then I have mite issues. And life happens. So sometimes that's the reason why I don't follow my routines. So again, too many inspections. I guess for me, 10 days is good. But for a beginner, it's better that you do more often than not. So you get experience with your bees to listen and smell them, to hear them. It's really important. And waiting to requeen. So learning to assess your queens is really, really important. And the sooner you learn, the better, because our seasons are short. So you need to requeen as soon as possible. So Sylvain, I'll answer your question about how do I transfer nurse bees. I'll answer that at the end. And so I'll tell you right now. So what you do is you take a brood frame, you shake all the bees off. You put an extra hive body above an excluder, above your brood box. And then you put your empty brood frame up there. Okay, so no bees and an excluder. So the excluder is to make sure the queen doesn't go up. Then what you do is you put your lid on and then come back in 2-3 hours. So in 2-3 hours, the bees that are on that frame, on that brood frame, are nurse bees, mostly. So probably 95%. So those bees have never really foraged. They've never really been outside. They're docile. They're not aggressive. So those are the perfect bees to transfer into another colony. So that's how you transfer nurse bees, is you take a brood frame, stick it above an excluder for a couple hours. And when you come back, the bees on there are nurse bees. And then I would take that and put it into a weak colony. and they're getting a boost of nurse bees, so raise, because they need to nurse bees to raise new bees. And then they're going to get new bees from the brood frame in the next probably 7 to, especially if it's cap brood, so they'll get a bunch of new bees in the next 7 to 10 days. Let's see. This is our typical cycle up here. So again, I adapted this from Randy Oliver's site, and I took what we do see up here. So for example, let's start in May. So usually in spring is where we would see chalk brood. Okay, I've seen it a bit later, but usually it's here. The colonies are weaker, they have higher stress levels, and there is a link between stress and chalk brood. So that's where you'll see chalk brood. There's not much you can do other than reduce the volume, make sure there's enough bees, make sure they have food, and keep them warm, because we do get cold nights and it's about keeping them warm. Like nosema is usually, it's a winter disease and early spring. And if you've got it, there's not much you can do. The best way to do is get those bees to ramp up again and to basically clean out those sick bees. If you've got feces, a living colony, and there's feces and like dirty frames and a dirty box, that's where having extra boxes comes in really handy. And basically you can just put them in fresh boxes and clean out the old stuff. Because bees are very hygienic, they like cleaning things. So if there's poop in the colony, they'll want to clean it. I've seen it because I've checked it. So feces with a colony that has nosema, there's nosema spores in the feces. So it's better to clean as much of it out as possible and to give them some fresh stuff. Then we get our June peak where all the native flowers up here go wild, and it's great. And then for us here, our dry season where it gets really hot, I guess relative hot, or 25 degrees Celsius, is usually mid to late July. It gets really dry, not much rain. So the bees might go nectar deficient, pollen deficient, and that's sometimes where you'll see EFB and sometimes PMS if there's stresses. And then you can see then the next cycle is Varroa, because they're peaking in late, probably in August to September is when Varroa is peaking. So that's pretty much our disease cycle up here, and when to look for. For example, chalk brood may pop up anywhere, AFB may pop up anywhere, but this is the general rule of thumb. So mites, viruses because of mites, nosema in the winter, chill brood. So if you're doing inspections and it's below 40 Fahrenheit, 10 C, and there's a lot of eggs and larvae, and you keep those frames up and you're taking too long to inspect them, basically you're killing that brood. So that's why you need to be chop. Don't take too much time while doing your inspections, because you might be the cause of that brood diseases. So, and it's just called chill brood. It's where you're chilling the brood and you're damaging the larvae, and the bees have to get rid of it. So chalk brood, I've never really seen sac brood up here. So sac brood is a viral disease and chalk brood's a fungal disease. Again, both of these are fairly straightforward. Again, if it's a severe infection, it's better to get rid of the frames where you see sac brood or chalk brood and give the bees some fresh equipment. I think we had a beekeeper last season get EFB up here for the first time, and I guess this year they're burning all their hive equipment and starting over from scratch. And not taking any chances with old equipment. EFB is treatable, but usually it's more stress related. So again, pollen patties, sugar syrup if you have to, helps alleviate a lot of the EFB issues. Here in the Yukon, wax moth is not really an issue. You might get it if you get a nook from down south that has some moths in there and some larvae. but they won't survive the first winter. If you keep your equipment in a storage shed, or in a warm place, then the wax moth may survive and damage all your equipment. So it's probably very important that you, I keep them in a cold shed, and cold being when it's -40 out, it's -40 in the shed. So most of my spare equipment gets frozen for most of the winter. But learn to, I think it was Randy Lamb over here, he had a nook and he noticed some tunneling from a wax moth. He knew how to identify it. He pretty much picked it, and that it pretty much eliminated the issue with the wax moth because they won't survive the winter here. Bears, just have an electric fence, don't mess around. And ants are easy to fix. Is make sure your stand has some legs. I stopped using petroleum jelly. I just use that Tanglefoot that you get at the hardware store. It's a really sticky thing that you stick on trees. I know some people use duct tape, reverse duct tape, and they make a collar around the legs. But last year I used Tanglefoot and it worked awesome. And hornets and wasps, just keep your entrances small. So I keep my entrances maybe 2 to 3 inches pretty much the whole season. I may have a top entrance on some of the honey supers, but I'd say only my weak colonies have issues with wasps, but I have a really small entrance in those cases. So this is an example of deformed wing. It's really, really sad when you see it, because you feel guilty. Okay, so I'll show you a video about acute bee paralysis virus, and you'll see that's a very, very sad video, because we do care about our bees. So here's a close-up of these mummies. And then that same one, and then you can see it's like shotgun brood, and it was getting pretty bad. So the person who shared these photos with me, he just threw that frame out, and because there's no point risking infecting other frames, and the colony recovered and they're fine. So the fall broods, again, anytime you see like melted larvae or discolored larvae or cappings that look really weird, don't ignore the problem. So the first time I thought it was EFB. I did the string test. So I used a matchstick in the hole. I mashed up the larvae and I pulled it up. But it didn't string. It wasn't stringy like that. There's plenty of YouTube videos that show you this. So I'd say just YouTube American foul brood test, and a bunch of them comes up. But it looked like my problem was parasitic mite syndrome. I actually didn't have any mites, but I guess there was a lot of residual mites from the previous year. So it does stick around with it. So I found this reference sheet really handy, and you can find it at this Texas bee inspection TAMU EduFile. So it was a really nice explanation about the different brood diseases and how to identify and what to look for. American foul brood, just burn your stuff. If you've got polys, you can disinfect it, but make sure you get into those nooks and crannies, okay? Because, but I'd say burn all your frames, and you can probably wash everything with very concentrated Javex or chlorine bleach to make sure you're killing everything. So this was my situation. So I had this, and you can see I had some larvae popping up. So the red it was like sac brood, and the blue melted larvae is like, it was more melted. It wasn't really chalk brood, but it was more like EFB. So it was weird. So then after sharing with a few people, it pretty much came obvious that I had PMS, because I was also finding deformed wings. So they had aborted some larvae and pulled them out, some pupae, and they had all basically deformed wings on them. So this was during one of our rare heat waves. So we had about 2 weeks of hot weather. So it actually got up to over 30 degrees, which is really rare up here, but it was super dry. And it was basically no nectar coming in and no pollen. So what I did is I threw a pollen patty and some feed, and about a week later, it worked itself out. So let's see if this video works. So this is one of my bees, and it was a colony last year. So it was a boomer hive that came out with like 16 frames of bees, and it was just awesome. But you can see there's a little mite on its back here, and she's dragging her tongue, a bunch of convulsing bees. A lot of people think it's poisoning. That's what I thought initially, because this was at the same time as, I guess, death camas was blooming. And I guess death camas is poisonous. The pollen's poisonous for most pollinators, except a few specialized bees. But you can see it's really sad. So I was getting it wasn't bad, but I was getting maybe 20, 30 funny looking bees every morning doing this. And I was like, what the hell's going on? And you can see there's a bouncing bee up there, and it was just sad. So, and they're all messed up. So once I learned that this was a Varroa related virus, then I said, okay, enough's enough. I need to follow my routine, because I neglected this colony in spring. I usually do a spring treatment, but this time I didn't do it, and I let it get out of hand. Okay, so, and again, you don't need to use miticides. There's other ways of treating mites, but they do require more effort. I've never used a miticide before other than oxalic acid vaporization. So this is just an example. This is Megan Mildbrath. I guess at Michigan U, she has a really good, it's called Planning for Varroa. If you want to learn about Varroa, she writes wonderfully, and she's a great resource. So I'd say this is probably your best guide to Varroa, and it goes through soft approaches, and it goes down to more, I guess, invasive type approaches. So this is one of her charts, and it just shows how it's exponential. So I crunched the numbers. I've got a couple of models that I looked at. And if you've got like a package, say with 400 mites, when you first get it, we'll have up to 20 to 30,000 mites come September. Okay, so it literally spikes perfectly late August, early September. But for example, if you treat that colony and you bring it down, say, especially a package, there's no brood, and you do an OEV treatment, and you knock out, you bring them down to 10 to 20 mites. So it'll go from 20 to 30,000 mites to a couple of hundred, if even that, going into winter. Okay, so that's how much difference it does make. So again, you can see how the mite cycle coincides with September, October, and it's this downslope is where this is bee population, the brood, the last brooding of the season is where they're producing their winter bees. So mite biology. Basically, they feed on the fat body. So Samuel Ramsey, he's done a couple of really, really good talks. There's a group of scientists that did some research, and they figured out that the mites don't feed on the hemolymph, so the blood of the bee. They actually feed on the fat body. So fat body is like a liver for us, and it controls 9 main functions. So it affects basically how they grow and how they transform into a bee. So hence deformed wing and so all these different issues. How they store energy, how they detoxify for pesticides. The osmoregulation is how they manage water. Their immune function, how they thermoregulate, how much, I guess, the metabolic activity, so how they produce heat, manage heat, the conversion of sugars, how they synthesize protein and pollen into fat in their bodies. And then a critical one is that vitalogenesis. So it's like a lipid protein solution that's in the fat body that, because I guess in spring, late winter, there's no pollen. So the bees start brood rearing without pollen. So they're actually using their fat bodies, so this substance here, to generate and to brood rear the first generation of bees. So when we talk about winter and we talk about fat bees, we're talking bees that have a really healthy layer of this vitalogenesis, or not genesis, but vitalogen. I can't pronounce it. But if you Google half this word, it'll come out. So it's a substance that they need to brood rear. And if you have mites affecting the winter bees, they're actually reducing all these functions, but also affecting how much of this is actually getting put into reserves. And basically the queen lays an egg. The mite sniffs out the cell probably at day 8, just before it gets capped. So they're really smart. And then just as the cell is getting capped is when they go in. And then she lays a few eggs. So a male egg first, and then some female eggs. The male hatches first, and the females comes out, they get mated, and then the cycle continues. So then they start feeding on the larvae because it's soft-shelled. And then step 6 is at 21 days or 24 days, the bee emerges. And then you've got a bunch of new mites. So the Varroa sensitive hygiene. So what that is a lot of times the bees will sniff out the mite inside the cell and they'll break the capping. Okay, so by breaking the capping, they're affecting the atmosphere in here. And what it does is either completely eliminates the mite's capability of mating and raising new mites in the cell. And then a couple of days later, or a few hours later, they recap the cell, and it disrupts the whole mating cycle or the brood rearing cycle of the mite. So that's what that VSH behaviors is. So we'll start with integrated pest management, and this one's focused on mites. And some of these are in different orders depending what they're talking about. So first one is tolerance level. Up north, a lot of times our bees won't survive the winter if you do 0 about mites. Okay, so I'd say acceptable limit. If you've got neighbors and they have bees and you do nothing, for me, that's not an acceptable level. You need to do something to help control your mites. Okay, so here's one of those green drone frames. So technically you'd get them to draw out, cap it. And I used to give this to my chickens, but then it would take forever for them to draw it back out. But then I started freezing these frames, and then it kills all the mites and larvae. But now I'm reading that if there's viruses in these mites transferred to the larvae, then they can get retransmitted to the colony, because the nurse bees, the housekeeper bees, may get viruses through feeding. Okay, so this is an example of a medium frame. put into a deep. And you can see how the bees draw out a drone frame in the bottom here, or not drone frame, drone brood. And then you just scrape it off. And then you give it to your chickens and they get a really nice treat. And then you've got a medium, and it's a nice natural way. And a lot of times what you can do is you peel some of these cappings and pull out some of the larvae and have a look to see how many mites you have in there. Okay, because if you see mites in almost every cell, then you have a pretty severe infestation. If you don't see many, then good. I guess Sam's asking about CO2 in the hive. So that's an interesting topic, and it's, yeah, I'll speak to that at the end a little bit. I know when we use no top entrance when we're wintering, the CO2 levels will be higher in the colony, and it's yeah, so higher than normal CO2 disrupts the wintering mites. So it's another way of potentially managing mites without using hard treatments. So this cultural practice, I'd say it's all diseases, all pest management is, I keep a plumber's tool now with me. So I tend to burn or heat up all my hive tools after every use. I haven't gone to disposable gloves, but I plan on buying a box this year. And then you start inspecting with your healthy hives first. Okay, so if you've got 4 hives, you've got 1 booming hive and 1 weak hive, start with your healthy hive first. Okay, because you don't want to transmit anything the weak hive might have to your healthiest hive. Okay, so always start from the best hive and go down to the weakest or the sickest looking hive. And that's just a best practice to manage potential disease transfer. The other one, good nutrition. Again, make sure they've got feed and make sure they've got pollen when available. Let's see. Again, biological controls for me is genetic traits. So make sure you've got queens that do have some mite resistance or some hygiene behaviors for brood diseases and chalk brood and all that type of stuff. So physical controls, that's an example. So the drone frames and the mediums in a deep, screen bottom boards. Just from the natural drop, they say it's about 20% lower mites. So if you got 100 mites, 20 of them might drop out because of a screen bottom board and they can't get back in. And that is not enough, but it's better than nothing. So that in combination with a brood break, so making splits or requeening the colony are all good ways of managing mites. If you don't have many mites, these should be enough to manage your mite levels, okay? The caveat here is when you get your package, doing something early will determine if you can just do this. If you do nothing with your package, or you have neighbors that have mite infested colonies and your bees like robbing theirs out because they're just bee havers and they don't take care of their hives, then you'll have other issues. Okay, so, and then the other one is monitoring. So you can do an alcohol wash where you just put a bunch of bees in a modified peanut butter jar with a little screen, and then you put some alcohol in there and then you shake it, and then you can see how many mites wash off the bees. So I can share some videos on how to do that, but it's fairly straightforward. And if you don't want to kill any bees, you just use powdered sugar, and you take about, again, a half cup of bees, you throw in a couple tablespoons of powdered sugar, and then you shake like crazy for about 2-3 minutes, and you've got to shake really hard with powdered sugar to really get those mites knocked off. So that will tell you how many mites you have. So my monitoring, the first monitoring of the season, I just do an OAV treatment because my mite counts are generally fairly low. And my mite drop is low, and I do alcohol washes, it's very low. So I get 1 to 2 mites max, usually 0. So I found I just do an OAV and it tells me right away, do I have mites? Yes or no. And it gives me a better number of how many I have. If anybody in the Whitehorse area wants an OAV tool, there's somebody selling there. So just send me an email and I can get you in touch with them. And the chemical controls. So soft is your oxalic acid vaporization or dribbles. I think there's some essential oils, so like Hive Alive and stuff like that. But those are really soft. OAV is semi-soft. It'll kill the phoretic mites. So phoretic is the mites on the bees, but it won't affect the mites in the cells. A bit harder chemical is formic acid. So Formic Pro is these little pads. It's a bit more finicky and you will end up damaging some brood. And it does disrupt the brood cycle. So it's a bit harder on the bees. And you have to be more careful of when you do it and watch the temperatures. So I have a package of treatment, but I've never used it. And I'm still debating if I'm going to use it or not. Because currently the way I'm doing it, if I follow my process, it does work. If you want to do Amitraz and all those other things, that's up to you. I've decided I don't want any pesticides in my colonies. I'll stop at the soft acids. So this is an explanation of how you do an alcohol wash and then how you do a natural mite drop. So this is from a couple of weeks ago, actually 2 weeks ago. So the natural drop, you can see was 0 in most of them. I had one that had 5 mites and the other one had 1. This was the relative strength of the colony. And then on May 9th, I did a treatment. Okay, a single treatment. And over 3 days, you can see how I have one colony at 77 and I've got one at 47. This one here was broodless. Okay, this is the one I just requeened and the queen's dead. So now it's a I think Alberta AB21. Okay, so you can see how most of them are in the green, but the yellow ones and the red ones, I'll do a retreat because I want to bring that number down. And I may just do a full other treatment. So I'm collecting data to see what the threshold should be. And this is just an example, kind of hard to see, but all these little black dots there, those are all mites. So this one here, there was probably in the 500 range. I count a few hundred and then I say enough, and then I just put a number to it. Because anything more than 100 is too many. And you can see this is my treatment last August. And you can see I had some high numbers here. And for example, hive number 2, I retreated, see how the June, so I didn't do an OAV until June. I had about 70. And this one here, I actually did about 5 treatments over and over and over again. And I was getting consistent. So overall, there's probably a good 5,000 to 6,000 mites in this colony. And this is also the one that had that video I showed came from this colony here. So now I will treat in May, early May. It gives me an idea of how many mites I have through my screen bottom board. Anything that's over 10, I'll do a retreat. This 8-1, I might just do it because it's there. But, and then I'll get an idea of the numbers. And the goal is to do it every 5 to 7 days until the number is below my threshold. Okay, so over here, there's a really interesting website. And what it does is it gives you your estimated, I guess, mite infestation level based on your OAV drop. Okay, so he does a count at 30 hours, and then he has a calculator to predict what your percentage is. And usually anything above 1 to 2% now is, they say, treat. Because you can see how 11 mites became a few hundred. Okay, so remember, these are phoretic mites. So probably a third, I forget what the exact number is. So 100 mites on no AV drop when there's brood is equivalent to probably 1,500 mites total in the colony. Okay, so same with here. This one I did multiple, so it was in the thousands. So there's a poop load of mites in this colony. Luckily, this H6, there was 200, but it was broodless when I did the treatment. So there's a good chance that I got 95% of all the mites in that treatment. And now you can see there's only 5 mites in the spring. So the treatment of this one actually worked. But you can see there was 70 left here, but right now there's 77. So this was a boomer hive. This was my strongest hive last year. And it was strong hives, lots of mites. Okay, so we're almost done. I do have new slides, but I guess I forgot them on my other computer. So they were easy to read. But this is one of those models. I'll answer that question in a minute. So the do nothing with a package. So study shows that you have probably a mite to 5 or 6 mites per 100 bees in a package. So this is the research paper that they were looking at. They were trialing using oxalic acid dribble to treat the packages. But I think OAV works, it's easier. I've found it easier to do. And if you use poly hives or even wooden hives with a solid bottom board, I just treat OAV through my screen bottom board. I pull the tray, and that way I don't damage the styrofoam. But anyways, you can see how there's 20-some thousand mites in September, late September. And again, you got your winter bees, so your brooding winter bees are being affected. And then the winter bees left over, the last population here is what gets, each one gets associated pretty much with a mite. So this one here is do nothing with an overwintered colony. And you can see it's still high. Yeah, 7 frames overwintered. This link here, I'll take you to this tool. It's a guy on Vancouver Island who developed it. So you can put your start criteria, your laying rate, if you do treatments or not. It's actually a neat tool. And then it gives you your potential load. Okay, and then do something pretty much if you treat twice. So if you treat in May and then you treat again after you take your honey supers in mid-August, pretty much your mites aren't an issue. So here it says 4.6. Technically it's high, but there's screen bottom boards, there's IPM, there's all these other things you would do that would help reduce the number even more. Okay, so do nothing. And this is literally nothing. So no IPM, no nothing. If you do IPM, it helps. But if you do something, especially when you get your package, okay, you get your package over here. If you do something early, you've just set your curve up for good. Okay, so it means that probably the following year, again, you just do minor IPM, maybe 1 treatment to see what your mite levels are in the spring. If they're low, you do nothing. And that's my motto is if I don't have mites, then I don't need to treat. If I have mites, then I need to do something. And to Joe's question, so Let's see. Okay, OEV right now, you do it without honey supers on, and that's the regulation in both Canada and the US. Chances are in the next year or two, the regulation may change because there's some testing going on now that the natural levels of OAV, or not OAV, OA, so oxalic acid in honey, the baseline and the baseline after treatment is the same. It doesn't really change. So it may change. Technically, the impact on the bees is really low. I have a respirator with the chemical cartridges, so the pink cartridges are for organic pollutants, and so the 3M mask. I wear a 3M mask and I stand upwind when I do it. And the way I have it set up is I can do it at a distance. So for the human aspect is, it's more when you're doing the treatment, you don't want to breathe in these vapors because they'll damage your lungs. So hence you need PPE. Some people just do it downwind, or they treat downwind and they stand upwind. But I guess in my line of work, I have PPE, so I use my mask to make sure I stay safe because I don't want to damage my lungs. Even though the risk is minor, if you follow carefully, but I'd say you take no chances, just wear your PPE, and that way you avoid any issues. And the masks, I just have a half mask and I wear sunglasses, safety glasses. So it's a fairly easy, once you get a routine, you see how it goes. It's low risk if you follow the procedure. Hopefully that answers this question. And again, so this is the tool I have. So it's a little metal, I think it's a steel cup connected to a DC battery that superheats this this little cup, and it superheats the crystals into a vapor, and then you seal the edges of your colony. I have a piece of plywood so the vapors don't go down. I seal the entrance and I run it. Here it says it's a gram. They give you a little spoon there, a gram per box. And then they say to run it for 2.5 minutes. So if I run it for 2.5 minutes, there's still some crystals left. So I run it for 3 minutes, and it vaporizes the whole gram or 2 grams. And I've got electric fences with 12-volt batteries in all my yards, so it's easy to treat. I just use my electric fence battery to run the tool. Okay, and this is an example. So you can buy cardstock that's specific with this grid, or you can make your own, a piece of white cardboard, old file folders, and you cut it. If you don't have a screen bottom board, you can just slide it through your bottom entrance after you've treated, and then you put your entrance back on, and then you can count. You just pull it out every 24 hours or every, say, after 3 days, and then you could count how many mites got killed during your treatment. Because a lot of people will use treatments, but then they don't measure how efficient the efficacy of the treatment. So they don't really know if it worked or not. Okay, so, and for me, it's important. So if I'm going to treat, I want to know it worked. And then if I retreat, again, it's to bring this down to nothing. So this was that hive number 2 from last year. So I got this like 5 times before I got the mites under control. So that's me doing nothing. And pretty much that colony collapsed. And then it had viruses. It's alive today because I treated, and it's ramping back up. There's no more signs of viruses. Bees are healthy. It's a growing colony. It's the same queen I had last year, but pretty much there's no more mites, so she's doing way better.